Hello. The topic for today's session is the biology and psychopharmacology of depression. My name is James Kalat. I'm a professor at North Carolina State University. Let me start with one disclaimer, which is that I am not a clinician. I do not do research. My knowledge of this comes from being a textbook writer. I try to keep up to date with the literature, but I'm not speaking from personal experience. Okay, so with that out of the way, these are the topics that we'll be covering: genetics, the drugs, and what we think we know about how they work, some of their side effects, and then the pros and cons of using psychotherapy or using the pharmacotherapy. Depression is certainly very widespread. A very large survey of people found that 5 to 6% of American adults reported depression within a given year, and 16% at some time during their lives. Don't take those numbers terribly seriously. Depression, like anything else, varies in degree from severe, no doubt about it, to relatively mild. And although we try to have consistent criteria, certainly some people apply those criteria differently from others. There's going to be a lot of marginal cases that one person would call depression, somebody else might not. And I will show you some other statistics on other slides which will contradict what I just did. In fact, the very next slide, this shows the relative prevalence of depression across a number of countries. And if you look at the United States, this survey showed slightly more than 10% of people in the United States reporting depression. So that could be different criteria. It could be just a different part of the country or even a different year. All these things make a difference. And you'll notice India reported more than 30% depressed within a given year. Is depression really that common in India? I don't know. The alternative is that maybe they are applying their criteria a little more loosely. Now, I don't want to dwell too long on the symptoms. I assume that the basic phenomenology of depression is familiar to my audience. But I do want to emphasize several points. One is that a number of recent studies say that the main emotional feature of depression is not increased sadness, but decreased pleasure. For example, if you ask people to keep track of all the events of the day and how they felt about it, depressed people don't report any more sad events or feeling sad any more often than anybody else. The big difference is that they don't report being happy. In fact, they hardly ever report being happy. And another study examined how people reacted when they looked at pictures. And they found that depressed people did not look any different from anybody else when they looked at sad pictures, but they failed to smile when they looked at happy pictures. So you can see how this could be kind of a vicious circle where people who can't enjoy anything, can't even imagine enjoying anything, become inactive. And if they are inactive, they are less likely to do any of the things that might bring some joy or cheer to their lives. Okay, another symptom that's worth emphasizing is sleep disorder. And you'll see on this next slide, on top, the typical sleep pattern for a normal person going to bed at the normal time of day, where they have a prolonged stage 1, then 2, 3, nice long period 4, then 3, 2, long REM period. Down below, you see the typical pattern for a depressed person, where he or she goes down through stages 1, 2, 3, 4 very quickly and back up to REM much more quickly than normal, and then might wake up and have trouble getting back to sleep. Now, that is a pattern that you would not see in a normal person, except if that person were going to bed much later than usual. If you or I went to bed at 3 or 4 in the morning, we would show that same pattern. And one interpretation of depression is that they are showing a problem with their circadian rhythm, that the circadian rhythm is phase advanced. It would be as if the person had just moved several time zones to the west and is going to bed at a different time of their cycle. Now, I'll come back to that way at the end. For the features of the sleep disorder, the short latency of REM, high density of eye movements during REM, awaken early, can't get back to sleep, feel a little groggy during the day. And all these features precede the other symptoms of depression. Typically, the person shows the sleep problems before they show other symptoms. And a longitudinal study of adolescents found that adolescents who reported major sleep problems were significantly more likely than other people to become depressed within the next 10 to 12 years, which then says that this is not a reaction to depression, but this is like a marker of who is vulnerable to depression. And it opens up the possibility that we could monitor people's sleep problems, identify those people who are maybe more likely to become depressed, and at least think about early interventions to prevent depression. One final thing about symptoms is very important for the research on effectiveness of treatment. Depression occurs in episodes. That is, the person is not permanently depressed for the rest of their lives. By definition, an episode would last at least a couple of weeks and typically lasts a couple of months, sometimes lasts a year or more, but not forever. And people will show this kind of spontaneous recovery no matter what you do. Now, the consequence of that is if you test the effectiveness of some kind of treatment and you find that over a course of time some large percentage of people get better, that doesn't mean anything unless you compare it to how many would have gotten better on their own. You need a placebo control group or a passage of time control group to find out whether your treatment worked at all. A couple of other things. People who are becoming depressed for the first time usually report a major stressful event. They'll say, okay, I became depressed because I lost my job, my marriage broke up, a close relative died, some other big stressful event. Okay, after later stressful events, they usually don't report or are much less likely to report a stressful event. And the later episodes come more frequently, generally briefer. It's as if the brain is learning how to become depressed and it's getting better at it. The first one, it requires a stressful event to get there. After you've been depressed, it gets easier. And the more times you've been depressed, the easier it is to get depressed again. Now, that is similar to a couple of other things. The same is also true for epilepsy. The more epileptic seizures you've had, the easier it is to have another one. The more times you've had a migraine headache, the easier it is to have another one. In each case, it's as if the brain is learning how to do something which we would prefer that it not do. Later episodes also tend to be more difficult to treat. And the message from that is it would be really nice to get people to get some sort of treatment right away. The first episode is easier to treat if you get the person to the therapist, but most people don't, certainly don't go immediately, and are much more likely to try to deal with it themselves and may not even show up at a therapist's office until they've gone through 2 or 3 sessions, at which point it's starting to get increasingly difficult. Okay, now what leads to depression? Well, guess what? Heredity and environment. There's good consensus that depression depends on, first of all, stress. The more stressful events someone has had, the greater the probability of depression. But there's also predisposition. Some people are much more likely than others to become depressed. Some people live through really horrendous experiences, and they're resilient. They bounce back. And within not too long, they're back on their feet. Other people go through prolonged depression after events that were really, would seem, no worse. So some people are more vulnerable than others. Genetics is certainly one of the first things we want to look at. It does tend to run in families. The more relatives you have with depression, the greater your probability. And this is another slide, which again, you're going to see different statistics. I've told you a couple other percentages of people with depression. This is a lower percentage. But this, at least here within the study, you can compare, because they're using the same criteria, and find that people who have a biological relative with depression are more likely to become depressed than those with an adoptive relative, but those too are more likely than those who have no relative. And that implies that you've got a couple kinds of factors. There's probably a genetic factor and a factor of whatever is in your family, whatever experiences are happening in families where other people are depressed. Well, over the last 20 years, biochemists have gotten very good at identifying individual genes, such as the gene for Huntington's disease. And in the case of something like depression, certainly a big effort has gone into trying to find a gene, if there is a gene. And the consensus has emerged that there is no such gene. There is no one gene that has a very big effect, and there's apparently no gene that has a consistent effect. Even a small effect would be interesting if you found it in every study. It doesn't turn out that way. It appears that there's probably a great many genes, each of which adds a little bit to the probability of becoming depression. The genes, however, are not specific to depression. It's not that there is a depression gene or even 100 depression genes, but the genes that increase the probability of depression increase the risk of lots of other things also. And it's worth noting that many or most of the people with depression have other disorders also, such as anxiety. People with a relative who has depression are at greater risk for depression, anxiety disorder, everything else on this list: attention deficit disorder, binge eating disorder, fibromyalgia, migraine, headache, PMS. I know that's not the official term, but that's what most people call it. So that says that you've got genes or other predispositions that make you more likely to break down in any of a number of ways. And which way you break down depends on something else. Very recent study has this interesting finding. Difference between people with early onset depression and late onset depression. Here, usually the dividing line for depression is about age 30. So if someone has a biological relative with early onset depression, somewhere in the 20s, they are more likely to have a variety of psychiatric disorders, depression, anxiety, and so forth. If you have a relative with late onset depression, the later the onset, the greater the probability that relatives will have problems with their circulation, that is, their blood. And that has some implications. It says that maybe we should be looking for more than one kind of gene. Maybe we should separate out two kinds of depression. Do one study on the genetics of early onset and another one on late onset. Also suggests maybe there would be different kinds of treatment. Perhaps, because we certainly find that not all people respond to any one treatment, maybe the people with late onset have a different kind of disorder. Maybe they would respond better to something targeting their blood instead of their brain chemistry. That at this point is speculative, but it seems like something that needs to be explored. Now, one of the most interesting studies pertains to the serotonin transporter protein. Let me explain a little bit about this. When a neuron releases its neurotransmitter, if that neurotransmitter is serotonin, dopamine, or norepinephrine, the transmitter will attach to its receptor, and then it goes back and gets reabsorbed through a process called reuptake, and that goes through a specific protein. In the case of serotonin, it's the serotonin transporter protein. And that will suck up much, certainly not all, of the serotonin. It's like a recycling system. It's to make sure that you don't have to keep making serotonin from scratch, which is a energy-intensive process and a diet-dependent process. So it makes sense to reabsorb it. Now, how much it gets reabsorbed varies from one part of the brain to another, and varies depending on how much of that serotonin transporter protein is present in the cell. Well, that in turn depends on some other genes. The gene for the transporter protein itself doesn't show any significant variation, at least not any common variation from one person to another. But near that gene, there are some regulatory sections of the chromosomes. Researchers discovered that of all the material on the chromosomes, only a small portion of it actually codes for RNA and proteins. And there's a lot that doesn't. And not that many years ago, they were referring to this as junk DNA, little pieces of DNA that were just along for the ride and didn't do anything. Now it appears that that was quite wrong, that those other pieces of DNA that don't code for anything modify the production of the other parts that do. And a portion right next to the gene for the serotonin transporter protein will increase or decrease the amount of its production. Well, this section of the chromosomes comes in two forms called S and L, meaning short and long. And the short form causes production of less of the protein, and the long form leads to more of the protein. Well, given the importance of serotonin, we believe, for depression, it would seem likely that that gene ought to have something to do with depression. Well, there were a lot of studies, some of which said that people with the S form of the protein were more likely to have depression, and a lot that found no significant difference. Well, in 2003, Avram Caspi published a very stimulating article, one of the most exciting articles published in the Genetics of Behavior maybe ever, in which he proposed that the reason it was not consistent from one stage to another is that the effect of the gene depends not just on the gene, but also on the person's experience. That the short form of the serotonin transporter gene predisposes to depression only if the person has a lot of stress. That is, it makes the person more vulnerable to stressful experiences. And therefore, the greater the stress, the more effect of that gene. And here's the graph that he showed. And what you see along the x-axis is the probability of depression. The y-axis, the probability of depression. What you see along the x-axis is the number of stressful events. And here he's talking only about big stressors, not something like I have a term paper due next week. These are big things like divorce, death of a husband or wife, loss of a job, and so forth, being physically attacked. So people who had no stressful events sometimes became depressed anyway, at least no stressful events that they were recording. Sometimes became depressed anyway. But it didn't matter for them which form of the gene they had. People who had one stressful event were a little more likely to become depressed, but again, it didn't matter whether they had the S or L form of the gene. As you get to greater and greater numbers of stressful events, 2, 3, and 4, the probability of depression went up and the dependence on the gene went up. So people with 2 copies of the S gene, of course you have pair of chromosomes. This is an autosomal chromosome. If you had 2 forms of the S, you were more vulnerable to depression, more likely to become depressed if you had a lot of stress. And if you had 2 forms of the L, then more resilient, less likely to react badly to the stress, and one of each, sort of intermediate. Absolutely fascinating study. This then inspired lots of other research. Maybe there's lots of other genes, the genes for schizophrenia or aggressive behavior or attention deficit disorder or whatever you might think of, might have their effects on people who had one kind of experience and not another kind of experience. Now, unfortunately, this has turned out to be difficult to replicate. And a review published in 2009 it reviewed a large number of studies that followed pretty much the same design that Caspi had done, found no overall link between the serotonin transporter and depression, that is the S and L form, and no interaction with stress, which then says the results don't replicate. Now, I think this is too interesting an idea to abandon that quickly. There's a possibility that there might be something to this. Anytime there's no correlation or an inconsistent correlation between 2 variables, the obvious possibility is that there really isn't any relationship between them. But the other possibility is that we've measured one of them poorly. If you have an unreliable or an invalid measurement of something, it's not going to correlate with anything else that you care about. Now, in this case, you and I are familiar with the difficulties of doing behavioral research. First of all, our measurements of depression are not bad, but they're certainly far from perfect. The measurements of stress are a whole lot worse. You count the number of stressful events. Well, okay, does pregnancy count as a stress? Well, for a 27-year-old married woman who is eager to have a baby, it's a stress, but it's nowhere near the stress it is for a 16-year-old who didn't want to be pregnant. What about loss of a job? Loss of a job for a 50-year-old who loves the job and takes pride in it and knows it's going to be hard to get another job. This is a devastating experience from which the person might never recover fully. Whereas loss of a job when you're 23 and you're still trying to figure out what you're going to do with your life, not that bad. All right, now, so you and I are familiar with the difficulties of doing the behavioral research. We tend to assume that the biochemical research is 100% objective, 100% accurate. So people can figure out whether you've got the S or L gene. It's not true. There are several methods of assaying the S and L gene, and the ones that are in most common use are prone to a significant number of errors. So we've got inaccurate measurement of behavior, sometimes very inaccurate measurement of genes. So how much of an interaction is there between them? I think it's too soon to say. The idea is interesting enough that I think it calls for more research. Now, this brings us to, I guess, the main topic, which is pharmacotherapy. Now, I understand that most people watching this are clinical psychologists. There's only a couple of states right now that permit some clinical psychologists to prescribe drugs. If you don't live in those states, why would you care? A couple of reasons. One is that you might be communicating with some physician who does prescribe the drugs, where you provide the psychotherapy and the physician prescribes the drugs. It helps to be able to communicate and to know what we're talking about. And if the doctor says something, you would like to be able to reply in a way that seems intelligent. Second, you might want to give advice to a client on choosing between psychotherapy and pharmacotherapy. And occasionally, you might be alert to drug interactions. A few years ago, I was contacted by an elementary school teacher who had a very troubled little girl in her class who was taking a wide variety of drugs, and the teacher was worried that the drugs might be more of the problem than the solution. And the list of drugs included Ritalin, which is a common drug given for children, and an antipsychotic, which is not commonly given for children. And it seems to me very likely to interact badly with Ritalin. Ritalin increases and prolongs the amount of dopamine in the synapse. Antipsychotic drugs block dopamine receptors. So the two were fighting it out. They had contradictory effects. So you take both of them, they cancel each other out, leave the child only with side effects. So if you have somebody who's taking a bunch of drugs, you may not know what all the drugs do, but it's an easy enough thing to look up online or in a good reference book and check whether this combination of drugs makes sense, or at least clearly doesn't make sense. Now, the first antidepressant drugs were the MAOIs, monoamine oxidase inhibitors. These were discovered by accident. In the 1950s, no one at the time anticipated that there would be such a thing as an antidepressant drug. This was before the days of any drug that affected behavior other than tranquilizers. And at first, nobody knew how they worked. But as they accumulated a list of drugs that seemed to work as antidepressants, they looked for what they had in common, and all the early ones were MAOIs. So here's a list of some of the ones that are still in common use. Monoamine oxidase is an enzyme found within the presynaptic cell, that is the cell terminal where it's getting ready to release neurotransmitters. After a cell releases its neurotransmitters, such as dopamine or serotonin, this gets reabsorbed by the cell. Sometimes a lot of it gets reabsorbed, and the cell may be producing a lot of it. MAO is an enzyme that puts the brakes on it, prevents the cell from maybe building up too high a level. If you have too much of one of these transmitters, it's possible for that to cause damage. So that's the function of MAO. MAOIs are inhibitors of that process and therefore would increase the total amount of those neurotransmitters and increase their effect. So you see in this slide on your left, the cell ordinarily releasing its neurotransmitter, it attaches to the receptor, goes back up into the presynaptic terminal where the MAO breaks it down. On the right, it goes back and the MAOI has inhibited that enzyme, so less of it breaks down and you end up with more. So at first it looked like maybe depression is this ridiculously simple thing. It's just people with depression don't have enough serotonin. And wouldn't it be nice if it were that simple? But as we will talk about later, it doesn't look like it's that simple. here just showing a little bit larger version of the same thing: release, reuptake, and breakdown by MAO. Now, the MAOIs are not commonly used anymore. It's not because of difference in effectiveness. As far as we can tell, they're about equal in effectiveness to the other types, but they have a lot of side effects. Side effects that are regarded as very unpleasant: drowsiness, either constipation or diarrhea, nausea, stomach upset, either low blood pressure or high blood pressure. People have to be very careful of their diet. You can't eat grapes or anything made from grapes, including raisins or wine and so forth. So MAOIs are almost never used today as the primary approach to depression. Sometimes if somebody doesn't respond to one of the other drugs, they might shift them to an MAOI. Now, second category is called tricyclics. That's based on their chemical structure. They have 3 little cyclic sections of carbon compounds. And here's a list of some of those. There's quite a large number of tricyclic drugs that are available. Now, what tricyclics do is to block reuptake. Remember this transporter protein that will be responsible for reuptake of serotonin and norepinephrine, or norepinephrine would be a different one for norepinephrine, a different one for dopamine. The tricyclics block the reuptake of serotonin, norepinephrine, and sometimes dopamine also. And here you see a little diagram of that. On the right, the normal pattern. On the left, the normal pattern. On the right, the cell releases its neurotransmitter, which then goes back to be reabsorbed, and it can't get in. Now, there's a number of other drugs that work on that principle. Cocaine works on that principle. It's more specific to dopamine, but what it's doing is blocking the reuptake of dopamine. Ritalin also blocks reuptake of dopamine. Now, the difference among these is partly which transmitter they block, and more importantly, the quantitative aspects of how fast, how long, how much they block. Now, the tricyclics, in addition to blocking reuptake, have a variety of other effects listed there. They block one type of glutamate receptor, a couple types of histamine receptors, etc. So this is what's called sometimes a sloppy drug. It's a drug that doesn't have one clear effect. It has a bunch of effects, and that makes it hard to figure out exactly what's relevant. All right. Now, the tricyclics also have side effects. Side effects which are sufficiently unpleasant that people usually don't want to continue taking them after they recover. And this is one of the problems, that if somebody recovers while taking tricyclics, they usually don't want to keep on taking it. You've got dry mouth and drowsiness, impaired sexual arousal, a bunch of other things that are really unpleasant, and that means that people who recover while taking tricyclic drugs usually don't want to continue after they get better, and therefore you have a significant chance of relapse. You could have decreased the chance of relapse by continuing to take the drugs, but people don't want to do that. The SSRIs have become very widespread and a very big business for the pharmacology industry. These do the same thing as the tricyclics, except that they are pretty much limited to serotonin. They don't have much effect on norepinephrine. They don't have much effect on dopamine. Prozac was certainly famous from the start. Citalopram, Celexa, the other ones listed there, are all among the most widely prescribed drugs in the United States today. The side effects here are more limited than they are for the tricyclics. The most common is nausea and headache, which is especially intense at first, but tends to decline over time. And a few other side effects which are less common, but worth knowing about. And some other things. There are now selective serotonin and norepinephrine retake inhibitors, and atypical, which just means they didn't fit any of the other categories. We didn't know where to put them. And of these, the one that's maybe most interesting is Wellbutrin or bupropion, which in addition to being a good antidepressant, also helps people who are trying to quit smoking. And who would have guessed that? Controversy. Not that many years ago, there was a report published that said that people, especially adolescents who are taking antidepressants, especially the SSRIs, had an increase in suicidal thoughts. It's worth mentioning that those studies never actually reported an increase in suicidal acts, just suicidal thoughts. But based on that, the FDA put one of the little black box warnings on all those antidepressants, saying that you should probably not give this to adolescents. And since then, the number of prescriptions for adolescents, prescriptions of antidepressants for adolescents, has in fact gone way down. Now, did that lead to a decrease in the suicide rate? The answer is no. If anything, the suicide rate has increased, perhaps because we are less effectively treating depression. So this is a controversy that needs to be explored. All right, now if you wanted to choose among the various antidepressants, how would you do it? The research says that they are all about equal in effectiveness. I'll show you a graph about that in a moment. They differ in side effects, so most people don't want to go with the MAOIs because of the nasty side effects. But in terms of probability of getting better or amount of probable improvement, there's not a demonstrable difference. I mean, obviously, I'm not going to defend the null hypothesis and say there is no difference. What we're saying is that the differences are so small that you can't be sure of measuring them with the moderate size of samples that are practical to use. There are a couple of studies saying that maybe there are advantages for clomipramine, venlafaxine, and escitalopram for moderate to severe cases. I'm skeptical. It may be true, but it's demonstrable that if you send something in for publication saying, I found a difference, it's easier to get that published than to say I didn't find a difference. So there could be other studies who didn't find a difference, and until we've got enough replications, it's hard to be sure. Still, if I were the therapist and I were prescribing a drug, I would start with one of those just because there's no reason to do something else. Maybe this is right, but I'd do it without any real conviction. Now, this is a graph, and this requires some explanation. This is a series of 4 different kinds of studies. In each case, you've got different experimenters. There's different criteria for recovery. There's different durations of the study. So you can't compare the MAO lines to the TCA lines and so forth. All you can do is compare within each line, because within each line, they're all comparable. They've all got the same kind of information. So, for example, under others, which is mostly the atypicals, you see they're a very high, almost ridiculously high placebo response. I would infer that a lot of these studies were very prolonged. The longer you wait, the greater placebo response, just because all depression comes in episodes. If you wait 2 years, almost everybody is going to recover on placebos. Okay, but in each case, let's say, for example, monoamine oxidase versus comparison. So they were doing monoamine oxidase versus any of the other kinds. You see very little difference. Tricyclics versus any of the other kinds. Again, certainly better than placebo, but really no difference between tricyclics and the others in terms of percent response, probability of recovery. SSRIs, no significant difference between those and comparison drugs, and the others, again, none. So this implies that the differences among drugs are very small. Small enough that it's going to be hard to be sure of them. You might as well choose on the basis of side effects. Okay, now, what about switching? If you don't respond to one drug, might you respond to another drug? Well, I'll tell you about the studies. Okay, you start off with people who are on a particular drug for typically about 3 weeks. They take the people who didn't respond and then switch them to a different drug for 12 to 14 weeks. This is a before and after study, no control group. They find that many of the people who didn't respond to the first drug do respond to the new drug. So where did these people learn about experimental design? If you don't have a group that continued on the first drug for that same period of time, we have no idea whether it was the change to the other drug or simply the passage of time, and they would have done just as well if they'd stayed with the first one. And in fact, there are other studies that compared switching to a similar drug, so switch from one tricyclic to another, which works practically the same way, or switch from a tricyclic to an SSRI or an MAOI. Switching to a similar drug was just as good as switching to a very different drug, which suggests that switching was irrelevant, that it was simply passage of time. Frequently, when somebody doesn't respond to a drug, the therapist will try increasing the drug. Now, here the studies were at least done right, where you have people who failed to respond to a drug, and now they either continue on that same dose or go to a higher dose. And here the results are remarkably consistent, that moving to a higher dose does not increase the percent response. So giving them a higher dose increases the side effects, but it's not therapeutically useful. There are a couple studies that suggest that adding a second drug, so you have now 2 drugs instead of 1, that might be better. Emphasis on might. Let me mention briefly something about St. John's wort. St. John's wort is an herb, and therefore not regulated by the FDA. You can go down to some health food stores and buy it off the shelf. Doesn't require a prescription. It's generally less expensive, and has become popular for people who want to treat their own depression. How effective is it? Very mixed results. There's some studies that make it look like this is comparable to antidepressant drugs or psychotherapy. Some that say it's a lot less effective. But there's a side effect that is easily overlooked. People don't usually notice any side effects at all, so they think this is a very safe drug. But we all have within our liver an enzyme that breaks down plant toxins. All plants make chemicals that are toxic to animals. That's their way of discouraging us from eating them. Evolution in action there. Okay, and we have enzymes that break down those plant toxins. It's a very all-purpose enzyme that breaks down a whole bunch of things. Well, breaking down plant toxins is a good thing, right? And St. John's wort actually increases the activity of that enzyme. So far that sounds like it's good. But remember, this is a very all-purpose enzyme. It not only breaks down a wide variety of plant toxins, it also breaks down virtually all medicines. So that if the person is taking St. John's wort in addition to antidepressant drugs, it's probably canceling out the antidepressant drug. Would also cancel out cancer medicines, AIDS medicines, wide variety of other things. There are anecdotal reports of women who were taking birth control pills and then took St. John's wort and got pregnant and couldn't figure out how that happened. And the guess is the St. John's wort decreased the effectiveness of the hormones in the birth control pills. So this is not a risk-free procedure, especially if the person is taking any other kind of medication. Now, this brings us to the biggest theoretical problem here. Remember I said that initially it looked like maybe this was really simple, that people with depression just didn't have enough serotonin. And all these drugs increase the amount of serotonin in the synapse, one way or another. And some of them also increase norepinephrine or dopamine. The problem is the time course. If somebody takes an antidepressant drug, this will increase the amount of serotonin in the synapse within minutes to hours, depending on the drug, but it doesn't produce demonstrable effects on behavior until the person has taken the drug for usually 2 weeks or more. So there's got to be more to the story. If we're just a matter of increasing the activity at the serotonin synapses, you ought to get rapid effects. For comparison, for example, with Ritalin, the time course works perfectly. We know that Ritalin reaches its peak of activity in the brain in terms of blocking the reuptake of dopamine about an hour and a half after somebody takes the pill. It produces maximum effect on behavior an hour and a half after somebody takes the pill. Because they match, we have good confidence that we understand what's going on. With antidepressant drugs, they don't match. They aren't even close. So something else has to be going on. What? And there have been several hypotheses in the past. So I'm going to tell you the current hypothesis that is dominant, but it's possible this one isn't right either. Okay, now, people who have depression will show the following features. There's shrinkage of the neurons in the hippocampus. Of course, you can only see that in people who've died and gone to autopsy, and you look at those cells, but you see that within the hippocampus, the average size of the neurons is smaller. Within the hippocampus, you make new neurons. This is something we didn't used to know. It was discovered within the last 20 years that you do make new neurons within the hippocampus, not in most of the rest of the brain. And people with depression make fewer new neurons. They also are deficient in forming new neural circuits within the hippocampus. Forming new neural circuits is important for learning, of course. Now, depression is also associated with a decrease in BDNF. That's called brain-derived neurotrophic factor. A neurotrophin is a chemical that promotes growth and survival of neurons. Literally, it means neuron food. And for example, in the developing embryo, the cells in the spinal cord send axons out to the muscles, and anything that makes a synapse with a muscle gets nerve growth factor, little neurotrophin from the muscle, which keeps that cell alive. Anything that doesn't make contact doesn't get neurotrophin, and it has a built-in cell suicide program called apoptosis, A-P-O-P-T-O-S-I-S. Self-destruction program. If you don't find a partner by a certain period of time, you kill yourself. Okay, so within the brain, there's also this BDNF, brain-derived neurotrophic factor, which is important for keeping cells alive and expanding them and helping them make new connections. People with depression have a decrease in BDNF. If we give BDNF to laboratory animals, this improves their learning and increases neuron proliferation. And it's been found that antidepressants, among various other effects, increase the production and release of BDNF. So BDNF is low in most depressed patients, increases as a result of treatment, and most studies, although not all, say that the more you increase the BDNF, the greater the antidepressant effect. It is also likely that psychotherapy also increases the production of BDNF. Which then brings us to the relative effectiveness of drugs in psychotherapy. I'm going to first talk a little bit about drug effectiveness and then drug effectiveness in comparison to psychotherapy. This is a complicated slide, but worth going through. What you've got on the y-axis is amount of improvement from 0 standard deviations up to 2. And along the x-axis is the initial severity, as measured by the Hamilton Depression Scale, where 16 is a very small elevation. The person would qualify as depressed, but just barely, up to 32, where the person is in deep trouble. Okay, so you see the red triangles represent the amount of improvement for people taking antidepressant drugs. And here they're lumping together all different kinds of antidepressant drugs. And the grayish circles are the placebo effects. Okay, so and the relative size of the triangles and circles represents how many participants were in each study. So at the extreme left, where we're looking at very mild depression, the response of the drug and the response of the placebo were identical. It says if you've got mild depression, there's no evidence that the drug helps you any more than the placebo. As you go across toward more and more severe, the average amount of response to the drug doesn't change a lot. They're showing it with a slight increase for intermediate levels of depression and then a slight decrease at the end. I'm not sure that would be taken terribly seriously, but overall you can say there's not a lot of change there as you go to more and more depression, but the response to the placebo drops off. So at low levels, the placebo is just as good as the drug. At severe levels, the placebo isn't going to do you much good. And there you start to see a significant difference between taking the drug and taking the placebo. This compares relative effectiveness of placebos at the extreme right, the medications, that is antidepressant drugs, and three kinds of psychotherapy. Cognitive behavioral therapy, interpersonal, which focuses on improving people's social relationships with others, and psychodynamic. This particular study, not very friendly to psychodynamic. There have been a couple of later studies where psychodynamic came out a little better than that. But what you can say is about 30% would recover with placebo. Now, how many obviously depends on how long the study lasts. This is a summary that goes over a whole lot of different studies with varying delays. But so, for what it's worth, about 30% recovered well with placebos, whereas those on medication, a little over 50, which means that medication was adding maybe another 25% to the response rate. The psychotherapy, about equivalent. The percent recovery was about the same. Now, this is with fairly short-term treatment in each case. If we extended that, we would get greater response to both psychotherapy and pharmacotherapy. We would also, of course, get greater response by the placebo group. But for what it's worth, the psychotherapy and pharmacotherapy appear to be about equal in effectiveness. Now, what would you guess as to how this interacts with severity? If you deal with relatively mild depression, which, if either, would be better, psychotherapy or pharmacotherapy? How about moderate depression? How about severe depression? What would you guess? Well, most therapists, certainly most psychiatrists, guess that the more severe the problem, the more you want to rely on drugs. The evidence doesn't bear that out. The evidence says that psychotherapy and pharmacotherapy are equally effective at all levels of depression, from mild depression, where in fact they're competing with the placebo effect, up to severe. The one exception on that is for dysthymia. Dysthymia is sort of long-term gloominess, pessimism. And in that case, the pharmacotherapy worked better. The authors of this review speculated on that. They said, here you've got people who've been gloomy and depressed so long, they hardly remember what it was like to feel better. Now, this is a short-term treatment. How much response are they going to get to psychotherapy within 6 to 8 weeks? It's probably not enough, whereas they might respond to the drugs. At any rate, for dysthymia, pharmacotherapy may be better. For standard major depression, either one seems to work about equally. And both of them increase metabolism in the same brain areas, including the hippocampus. That's why I said that chances are psychotherapy is improving BDNF just as much as pharmacotherapy. Some people find this surprising, but if you believe, as I do, that mind is simply another word for brain activity, anything that changes one should change the other. And since both of them are effective, it's not surprising that they might work through some of the same roots. Now, pros and cons. The big advantage to psychotherapy is that the benefits usually last longer. And I would like to come up with statistics to say what percentage of people relapse after various times on psychotherapy or pharmacotherapy, but it varies a lot from one study to another. I've seen percentage of people relapsing after 2 years after finishing psychotherapy, anywhere from 25% to 50%. And similar variation in percent of relapse after pharmacotherapy. What we can say, though, is, of course, they vary because each one has a little bit different criteria for what constitutes recovery and all sorts of other differences in procedure. What we can say is that every time anybody has compared psychotherapy to pharmacotherapy, people who recovered on psychotherapy had less chance of relapse than those who recovered on pharmacotherapy. unless the people in pharmacotherapy continued taking the drug, in which case they merely caught up to those who had psychotherapy. So in other words, if you had, let's say, 8 weeks of psychotherapy, responded nicely, you stopped taking psychotherapy, you've got a good chance of remaining symptom-free. You might relapse, but you've got a pretty good chance of long-term recovery. Whereas if you responded to the drugs, your chance of relapse is higher. A couple studies suggest that psychotherapy is better for people who had early traumatic experiences, such as a child who watched his parents get killed, some sort of really terrible experience. For those people, psychotherapy works apparently a lot better than pharmacotherapy. On the other side, drugs are cheaper, and the HMOs make a lot of the decisions. In a lot of cases, they won't pay for extensive psychotherapy. They might pay for 3 sessions, which is probably not going to be enough, or they could pay for the drugs. The drugs are more convenient. It's easier to pop a pill than to drive to the therapist's office and spend an hour once a week. And the drugs probably act a little faster. You get benefits within a couple weeks. Usually psychotherapy takes a little longer than that. Now, what if you take both? How much benefit is there? It's really hard to get a grasp on that. You would be able to combine the advantages, that the drugs probably act faster and the psychotherapy lasts longer. So you would have both benefits. But of course, that would be at increased expense. Does it increase the percentage of people who respond well? Again, there's some really bad research studies in this regard. There's one study that compares people who were taking a drug and didn't respond to it. Now they get randomly assigned, okay, so far so good, get randomly assigned to 2 groups. One continues taking the drug, the other takes the drug plus psychotherapy. Now, if those who had both did better, we'd be able to draw a conclusion. But in fact, it merely came out tied, which then says that the drug wasn't doing any good, but it doesn't really tell us whether psychotherapy was doing any good. Maybe those people just would have recovered by the passage of time. What they needed was a group that would be nice to have 3 groups from the start. One that takes psychotherapy, one that takes pharmacotherapy, one that takes both, and find out how much better they do. Now, there are a couple studies that indicate that there are some people who respond to psychotherapy who don't respond to pharmacotherapy. Apparently a very small percentage, a few percent. It looks like most of the people who respond to one would respond to the other. You've got about 30% of people who are going to recover on their own just by passage of time. Another 25% who are easy to treat. Give them either psychotherapy or pharmacotherapy, and within a few weeks they're fine. And then a bunch of other people who don't respond very well to either one. At least not quickly. What do we do about those other people? Electroconvulsive therapy is an option. Electroconvulsive therapy has a couple of advantages. It is quicker. It usually works within a week, sometimes 2 weeks. It helps a higher percentage of people. It helps about 90% of depressed people, including those who didn't respond to either psychotherapy or medications. One very big disadvantage, though, which is the relapse rate is very high. So somebody who recovers after electroconvulsive therapy is almost certain to relapse within a fairly short time. So the strategy would be maybe when all else fails, try electroconvulsive therapy. And then if the person recovers, now we've got to do something else to maintain their mood, either psychotherapy or drugs. Conceivably, repeated electroconvulsive therapy, although I think most people would be reluctant to try that. A couple other things. Exercise shows a lot of promise as an antidepressant, as a way of preventing depression and recovering from depression. Here we're not talking about anything strenuous. It's not muscle building exercises, just 30 to 45 minutes a day of walking, treadmill, elliptical walker, jogging, dancing, just something to get the blood flowing to the brain. Diet. Especially true for bipolar disorder, which we're not talking about today, but increase in omega-3 fatty acids, which are an important constituent of the brain, which you get primarily from seafood. Salmon is one very good source of that. Seems to decrease the probability of depression, and bipolar disorder across countries correlates very nicely. So countries like Japan that eat a whole lot of seafood have the least in terms of bipolar disorder, whereas countries that eat much less of it have more bipolar disorder. Light. For seasonal affective disorder, very bright lights for a couple hours a day works very well, and not sure why. There is some speculation that it might work also for people with regular depression, not seasonal. And altered sleep patterns. Remember back at the beginning I said that people with depression act as if they were had their circadian rhythms shifted back a couple hours. So if they go to bed early, then they get their full night's sleep and they wake up, and often that's a good antidepressant. But the problem with that is it doesn't last very long. So a couple weeks later, their circadian rhythm has shifted still more, and they would have to go to bed at 4 o'clock in the afternoon in order to get a full night's sleep. And after a while, that becomes impractical. But it says that if we could get really a good handle on sleep, maybe attack sleep as the primary problem, the depression might come with it. Okay, well, I think in summary what I can say is that depression is a widespread problem. There are a number of treatments that work some of the time, none of them that is perfect, and I hope that what I've had to say about depression hasn't been too depressing. Thank you.